I'm very delighted to introduce the next chair for the session. Dr. Anna-Lise Williamson is a professor of virology at the University of Cape Town, South Africa. She is a worldwide expert of HIV vaccine research. She is also a fellow at the Royal Society of South Africa and a fellow of International Society of Vaccine. Anna-Lise, please.
Thank you very much. Well, it gives me great pleasure to introduce Dr. Jacqueline Miller, who's going to talk to us about Moderna's coronavirus vaccine work. She's a pediatrician who for the past 20 years has worked in the field of vaccine development, contributing to the licensure of 5 vaccines. And she joined Moderna in 2020 as the therapeutic area head for infectious diseases and currently leads the clinical development and medical affairs teams developing Moderna's vaccines and monoclonal antibodies directed against infectious pathogens. So I hand over to you.
Thank you so much, Anna-Lise, and thank you to Shan, the ISV, and the meeting organizers for the opportunity to share with you the clinical development program for Moderna's mRNA 1273 vaccine. Next slide, please. So this is the standard forward-looking statement and disclaimer slide. Next slide, please. So the mRNA 1273 vaccine is a messenger RNA vaccine. It's a single sequence for the spike protein. There are 2 proline substitutions which keep that spike protein into the more immunodominant pre-fusion conformation. And this messenger RNA is dispersed in a lipid nanoparticle, which enables delivery to the antigen-presenting cells. So as explained by the previous speaker, the messenger RNA, once delivered to the cell, is then translated by the ribosome into its protein component. It is a full-length spike protein in pre-fusion conformation. It naturally assembles into its trimeric form, and it is cell surface expressed. The vaccine is given at 100 microgram dose, and it's given 2 doses 1 month apart. And the vaccine is given intramuscularly. So I'm going to talk a little bit more about how the dose was selected for this vaccine in the subsequent slides. Next slide, please. So before moving to tell you about the clinical development program, I'd like to summarize our preclinical data, which really enabled the ability to accelerate our clinical development. So we've investigated this vaccine in several animal models, including mice and non-human primates, and robust neutralizing antibody responses in both younger mice and aged mice, as well as non-human primates, have been observed to be induced. The vaccine also demonstrates robust protection against the lung challenge in mice and a pulmonary and nasal challenge in non-human primates. There's no indication of enhanced respiratory disease after a viral challenge, which was at 1.9 times 10 to the 5th, even when subprotective vaccinating doses of mRNA 1273 are used. And finally, a Th1 dominant phenotype of CD4 cells has been induced in both mice and in the HPs, giving reassurance about the lower likelihood of vaccine-associated enhanced respiratory disease. Next slide, please. So this slide gives you an overview of the clinical development plan. And what you can see is that the clinical development is being conducted in rapid succession. This is really being enabled by frequent interactions with the FDA and submission of both unblinded and blinded safety data as the program continues. I shared with you the summary of the preclinical data plan previously, and in the slides that follow, I'll give you an overview of some of the first in-human data in subjects who were 18 to 55 years of age. This was an NIH-sponsored phase 1 study. I'll then give you the overview of the ongoing phase 2 and phase 3 pivotal safety, efficacy, and immunogenicity studies. Next slide, please. So this is a summary of the NIH-sponsored Phase 1 safety and dose ranging study. There were multiple cohorts in this study in 3 age strata: a younger age strata of 18 to 55 years of age, an older adult cohort of 56 to 70 years of age, and an elderly cohort over 71 years of age. In each of these treatment groups, it was planned to investigate the vaccine at 25 microgram, 100 microgram, and 250 microgram doses. However, because of some grade 3 fever observed after the second vaccination in the 18 to 55-year-old cohort, we elected not to continue that dose into the older age and elderly cohorts, and instead are now also investigating a 50 microgram dose. All cohorts are fully enrolled, and the study is ongoing with safety and immunogenicity follow-up to continue for 12 months after the final vaccination. So the study population were healthy males and females who were at least 18 years of age, and we enrolled all subjects regardless of their baseline serostatus to SARS-CoV-2, although we have obtained baseline antibody titers and concentrations. Safety was followed through the completion of diary cards, and there were solicited adverse reactions captured for 7 days after each injection. A further unsolicited AE follow-up period for 28 days, and then SAE and medically attended adverse events will be followed throughout the study. Immunogenicity has been followed by looking at geometric mean titers of antibodies, seroconversion rates, and geometric mean fold rises. And what you will see in the slides that follow are the geometric mean concentrations and titers for both binding antibody and neutralizing antibody, as well as assessment of T-cell immunity. Next slide, please. So this slide summarizes the safety data that have been generated to date in the 18 to 55-year-old cohort. And what you see in the graph on the right, in gray are grade 1, in blue grade 2, and yellow grade 3 adverse events after the first vaccination and then the second vaccination on the right. For each of the symptoms listed, all 3 dose groups are given 25 micrograms, 100 micrograms, and 250 micrograms. The most common symptoms observed in terms of systemic symptoms are fatigue, headache, and myalgia. And this was observed more frequently after dose 2 than dose 1. In terms of local injection site reactions, pain was the most commonly observed reaction with minimal swelling and redness. No vaccine-related SAEs have been reported in the study, and no halting rules were identified by the Safety Monitoring Committee. Next slide, please. So now I would like to share with you the neutralizing antibodies that were elicited in this trial. And what you see moving left to right are the 25 microgram, 100 microgram, and 250 microgram doses in the younger age cohort, 18 to 55 years of age. This is a lentivirus reporter assay, and it's measured in assay titers at ID50. The pink range is actually a range of 41 convalescent serum titers that were tested for further comparison. The vaccinations were given at dose 1 and dose 29. And so what you can see is that by day 14 after the second dose, neutralizing antibodies within the range of convalescent sera were observed in all participants. The lowest antibody titers observed were in the 25 microgram group, with the 100 and 250 microgram groups being comparable. And the 100 microgram group, which was ultimately based on both the immunogenicity and safety results selected for further development, was in the top half of the range of the convalescent sera by day 43, which is 2 weeks after the second dose. Next slide. So I'd also like to share with you the CD4 T-cell data that were generated in this 18 to 55-year-old cohort. So what you see on the left are T cells, CD4 positive T cells that have a Th1 dominant phenotype. And this was measured in an intracellular cytokine staining assay that measured both interferon gamma, IL-2, and TNF alpha. And what you can see is that by 2 weeks after the second dose, there is a strong CD4 Th1 phenotype response with very little Th2 phenotype observed. And the Th2 phenotype was assessed by looking for IL-4 and IL-13. So again, these are reassuring data that helped us feel more confident into moving into later stages of clinical development in terms of vaccine-associated enhanced respiratory disease. Next slide, please. So now I'd like to shift gears and give you some information about the ongoing phase 2 and phase 3 studies. In the phase 2 study, which is sponsored by Moderna, 600 subjects are being evaluated in 2 age cohorts, a younger cohort that are 18 to less than 55 years of age, and an older age cohort over 55 years of age. Each of these cohorts will be vaccinated with either 50, 100 microgram doses of mRNA-1273 or placebo, and all of our cohorts are currently fully enrolled. Again, these were healthy subjects who were at least 18 years of age, and again, we enrolled subjects regardless of baseline serostatus. The safety endpoints that we followed were similar, and in terms of immunogenicity, we will be progressing with an ELISA assay and the pseudo-neutralization assay that I presented in the phase 1 data. Again, we'll follow these subjects for safety and immunogenicity 1 year after vaccination, after the second vaccination. Next slide, please. Oh, I think we need to go back one, please. Thank you. So now I'd like to review for you our pivotal phase 3 efficacy, safety, and immunogenicity study, which will ultimately enroll 30,000 subjects, and these will be randomized 1-to-1 to either receive mRNA-1273 at the 100 microgram dose on day 1 and day 29, or a saline placebo. And again, we have stratified the enrollment in this study in order to ensure that we are assessing patients that are at increased risk for disease. So first, we have a cohort that is greater than or equal to 65 years of age, and this cohort is being enrolled knowing that regardless of other underlying comorbidity status, these older adults are at increased risk of the severe complications of COVID. We also are enrolling a younger age cohort that actually also have comorbid conditions based on risk factors listed by the CDC in terms of having severe complications of COVID. And at least 25% and up to 40% of those 30,000 subjects will be in these high-risk groups. Finally, there is 60 to 75% of the study that will be enrolled in under 65 years of age without other underlying comorbidities. That group of subjects also is stipulated to either live in a geography or have an occupation or other risk factor that puts them at increased risk for COVID disease. And so by enrolling these 3 strata, we intend to demonstrate that the vaccine data are generalizable for the US population. So again, there will be approximately 30,000 subjects, but this is a case-driven study with 2 interim analyses built into the study, and vaccine efficacy will be demonstrated if the lower limit of the 95% confidence interval is over 30% and the vaccine efficacy is 50%. And these are aligned with the FDA criteria for the demonstration of safe and effective COVID-19 vaccines. Again, we'll enroll all comers with regard to serostatus, and in sub-analyses, we will look at the differing immunogenicity between people that are baseline positive versus baseline negative. The primary objectives are to evaluate the efficacy of mRNA-1273 to prevent COVID-19, and I'll share the case definition with you on the next slide. and also to evaluate the safety and reactogenicity of the vaccine, 2 doses given 1 month apart. In this study, we intend to follow both safety and efficacy for 2 years after the final vaccination. And this study is being conducted in strong partnership with the Operation Warp Speed. It's funded by BARDA, supported by NIAID and the COVID-19 Prevention Network. Next slide, please. So I wanted to give you an overview of the case definition against which the efficacy of mRNA 1273 will be measured. To be included as a case of COVID-19, a study participant must have at least 2 symptoms that are systemic symptoms, including fever, chills, myalgia, headache, sore throat, or smell and taste disorders, or at least one of the respiratory symptoms of COVID, including cough, shortness of breath, difficulty breathing, or clinical or radiological evidence of pneumonia. And the subjects must have a positive PCR nasopharyngeal swab, nasal swab, or salivary sample for SARS-CoV-2. While this is the case definition for our primary endpoint, there's also a secondary endpoint for severe respiratory disease. Next slide, please. And so finally, on the point of generalizability, I did want to talk for a moment to Moderna's commitment to ensuring that we enroll a representative American sample into this study. We have initiated a diversity and inclusion advisory committee to help us make sure that we are enrolling a diverse population of subjects that are representative of the people who are ultimately at risk from this infection. This Diversity and Inclusion Advisory Committee includes advisors from various groups in the NIH, including the National Institute on Allergy and Infectious Disease, the Operation Warp Speed, COVID-19 Prevention Network, Veterans Administration, and a faith-based initiative called Metro. It also includes the National Institute on Minority Health and the Office of Tribal Health. This group reviews enrollment, race, and ethnicity demographics on a weekly basis, reviews current outreach activities and outcomes, and ensures that if we need to make changes in terms of those outreach, they give us good advice on how to make sure we're connecting with those communities. Developing strategies to ensure the participation of individuals who are from communities significantly impacted by COVID-19, and supporting the development and implementation of retention strategies, again, because ensuring that we have strong safety and efficacy follow-up of the vaccine will be critical. Next slide, please. So in summary, the messenger RNA 1273 vaccine encodes the pre-fusion stabilized spike protein in a lipid nanoparticle which has been designed for delivery to antigen-presenting cells of the lymph node. The preclinical data demonstrated the induction of neutralizing antibodies, which protect against viral challenge in mice and NHPs. Interim data from phase 1 have indicated that a 100 microgram dose is generally well tolerated, with solicited symptoms mostly mild to moderate in severity and self-limited in duration. It induces neutralizing antibodies in the upper half of the range of convalescent serum with the induction of Th1 biased CD4 T cells. And finally, the phase 2 and phase 3 COVE study are underway. And next slide, please. Finally, I'd like to give a huge thank you to all of our collaborators. So there are many of them listed on the slide, but as I mentioned, the phase 2 and phase 3 are funded by BARDA. We've had the support of NIAID and throughout the development program, and the COVID-19 Prevention Network in particular in the phase 3 study. And I particularly want to call out our principal investigators and study site investigators and personnel, along with all of the patients who agreed to participate in these studies. They are the true heroes of this effort and initiative. So thank you very much, and I'll be happy to take any questions.
Okay, so thank you very much for a very interesting talk. First question is there are quite a few people asking about the stability of your vaccine, and what sort of cold chain do you need?
So the stability is actually continuously being investigated. So I'm going to present to you the stability and the storage conditions that are anticipated upon launch. The vaccine, once it's made available, will be shipped and stored longer term at -20 degrees Celsius. And then at the point of care, so pharmacy or clinical site, it will be able to be stored at 2 to 8 degrees for up to a week. And as I mentioned, the stability data continue to accumulate, and as we're able to extend those storage parameters, we will do so.
Thank you very much. Another question is, why do you get more effects as you vaccinate, adverse events as you increase the number of vaccinations?
So I think that that's probably a cause for speculation, as we haven't fully analyzed statistically why that might be the case. But if you noticed on the neutralizing antibody slides and the CD4 cell count slides, while there's a response post-dose 1, there's a stronger response post-dose 2. And in particular on the CD4 cell, you see that at day 43, a lot of interferon gamma is elaborated. So we believe that the adverse effect profile is aligned with the observation that the interferon gamma is induced. But obviously we will be gathering much more data in the phase 3 study and hope to have a more refined safety profile for the vaccine at the time of launch.
Okay, it remains for me to thank you very much for a really interesting talk, and we need to move on to our next presentation.
Okay, thank you so much.
So, our next presenter is Dr. Brian Ward from Medicago, which is a Canadian company. Dr. Ward received his medical training at McGill, University of London, Johns Hopkins, and his research training at Oxford, where he was a Rhodes Scholar, and Johns Hopkins. He joined McGill in 1991, where he's currently a full professor in infectious disease. He's been working with Medicago since 2004, and presently he's the Chief Medical Officer to help with Medicago's phase 3 studies of a quadrivalent plant-derived influenza vaccine. And today he's going to tell us about their plant-derived SARS-CoV-2 virus-like particle vaccine. Over to you, Brian.
So, Anna-Lise, thank you for the introduction, and thank you very much to ISV for the opportunity to present. I admit it's a bit of a daunting experience to follow Sanofi and Moderna. We're a much smaller effort, and we are a little bit earlier in our development, so bear with me. Next slide, please. The company is based in Quebec City with a production facility in Raleigh, North Carolina. We're currently at about 420 employees. And our focus is exploiting plants to develop vaccines and therapeutic proteins like antibodies using transient expression in these plants. And our vaccines are primarily in the form of virus-like particles. Next slide. And jump to the next one. This gives you an idea of the scale that we're working on. We have a pilot plant in Quebec City that currently has the capacity to make about 10 to 20 million monovalent adjuvanted vaccine doses annually. And our commercial facility in Raleigh, North Carolina can make, we hope, about 100 million monovalent adjuvanted vaccine doses annually. Unfortunately, if you go to the next slide, we're about 2 years too late to make a larger contribution to addressing the COVID problem. Because we are in the throes right now of building a much larger facility, about 10 times the size of our commercial facility in North Carolina, in Sainte-Foy, Quebec. This has a greenhouse about the size of 2 football fields, and when it is completed by the end of 2023 or early 2024 and certified, we'll be able to make, we think, between 500 million and 1 billion adjuvant doses of a pandemic candidate vaccine every year. Next slide, please. And go on one more slide. So the candidate COVID vaccine is based on our transient transfection technology, which starts from DNA and that the DNA is put into a defanged agrobacterium vector that we put into every plant cell by using a process called agroinfiltration, which is essentially immersing the young plants in a bath of these agrobacterium carrying the protein of interest, and then applying a vacuum to their roots. This allows the agrobacterium to enter into the vascular system of the plants. Takes about a minute, and then the plants are put into our greenhouses to grow for the next 4 to 6 days. If you look at panel 4, virus-like particles form spontaneously by pushing out to the cell surface and cause invaginations of the plasma cell, the plasma membrane. Running across from left to right is a cell wall, and on either side you have 2 plant cells. And you can see these large invaginations that form with literally millions of these virus-like particles that have bud through the lipid rafts. This means that the plant cells are actually intact, and we do not have contamination with DNA, RNA, and plant organelles when we digest away the cell wall. And this process actually results in the formation of these tiny virus-like particles that are between 80 and about 150 nanometers in size, which you see on the right by transmission electron microscopy. This works for influenza and COVID and a number of other viruses. Next slide, please. Why do we think that our plant-derived virus-like particles will work? Well, we have quite a lot of experience. Our lead product is lead products are virus-like particles for influenza, either monovalent for pandemic possible use and quadrivalent for seasonal use. And in vitro, these plant-made virus-like particles activate mouse and human monocytes and dendritic cells with morphologic changes consistent with activation. There's spontaneous release of pro-inflammatory cytokines, and there's rapid processing and presentation on MHC class 1 and class 2. In vivo in mice and ferrets, these VLPs are delivered very rapidly to the local lymph nodes where there's a marked increase in cellularity with activation of a broad range of mononuclear cells and production of a robust antibody and cellular T cell response with a Th1 bias. Superior protection to the split vaccine, even in very old mice, 20 to 24 months of age. And with the avian strain HA, which tend to be less immunogenic even in mice, we can get very good complete protection, in fact, even in the absence or very little antibody production. So we think that the cellular immune response is both robust and also effective against pandemic influenza strains. To date, in vivo in humans, we've conducted 15 clinical trials with our pandemic and seasonal flu candidates. And the data suggest a very strong safety profile in the more than 13,000 subjects who've received our quadrivalent influenza vaccine with good antibody responses and an excellent Th1 deviated CD4 T cell response. Next slide. In the context of the next, flip forward, please. I'm not sure where the animation happened. There we go. So we know a fair bit about our VLPs, and we have limited information about adjuvants. What we know from our animal studies with monovalent H1, H5, and H7 influenza vaccines is that we actually get very good dose sparing with alum and with an oil and water adjuvant GLASE. The cellular response was maintained, and we got excellent protection in the animal models with much lower antigen doses, so a very effective dose sparing strategy. We've also used alum and GLASE and AF in older humans with monovalent avian VLPs as well as quadrivalent VLPs. We didn't get much of a boost with alum, surprisingly, but we got an excellent improvement in the antibody response at a significantly lower dose with an oil and water adjuvant. So we believe that we will be able to actually generate good antibodies and a T cell response at fairly low dose with an adjuvant. Next slide, please. Next slide. So what are we targeting? Well, like everyone or almost everyone else, we're targeting a pre-fusion stabilized S protein that's been modified slightly to include a plant signal sequence We've abolished the cleavage sequence at the S1, S2 junction, and we have standard mutations to keep it in the pre-S, in the pre-fusion form. One twist is that we've actually used the transmembrane domain of the H5 influenza virus because it is very effective at organizing trimerization in the lipid rafts in the plant system. Next slide. All right, so the production in plants allows us to scale up. It doesn't require the use of live viruses. The very small size allows drainage, we hope, to into the lymphatic system, as we've shown in animal models with activation of antigen-presenting cells. And because these VLPs look very much like the virus, on the surface at least, we expect that they will be highly immunogenic. Next slide. And go on to the next one. Unfortunately, all I will be able to show you is the preliminary data, the early data in our mouse studies, but I need to show you why we are moving forward a little bit more slowly than we otherwise would. We've partnered with GSK. They are providing AS03. We've also partnered with Dynavax, who are providing CpG 1018. And our phase 1 study, which I'll describe right at the end, which is ongoing, is an attempt to find out which of these adjuvants is most compatible with our plant-made virus-like particles. These are both well-known adjuvants, and we expect that we will see either a balanced Th1, Th2 or a polarized Th1 CMI response with the help of these adjuvants, in addition to what we already know about the virus-like particle immune response. Next slide. So where are we? Our first safety and immunogenicity study in mice is largely complete, although we are still in the process of collecting a lot of the data. Essentially, we use 3 dose levels, 0.1, 1, and 10 micrograms, with and without AS03 or CpG. And if you move on to the next slide, 2 doses, 21 days apart. And you can see this is essentially the outline of the study, which has just been completed, where we're looking at the regular toxicity measures immediately and full histopathology, because this is our toxicity study. Next slide, please. What I can tell you is that at the current time, our safety results are actually excellent with all 3 of these formulations at all 3 dose levels. And the immune response parameters that we'll be looking at will be a standard ELISA against the spike protein, neutralizing antibody responses, which are in fact going to be run by Battelle, not MRI. We have a surrogate pseudovirus assay, which is being run by Nexcelis. I'll show you some of those data. And we also did ELISpots for gamma interferon, and I'll show you the very preliminary data that we have in the mouse studies in the next slides. So go on, please. So, like many other vaccines, we actually mount an excellent immune response. On the x-axis, you have the 3 dose levels, the plant-made virus-like particles by themselves in green, with AS03 in red, and with CpG adjuvant in blue. And you can see that while the VLP by itself does elicit a reasonable antibody response, we get a significant boost and dose sparing with the both of the adjuvants. If you go to the next slide, you'll see the neutralizing antibody responses after the first and second doses. And these responses are even more clearly enhanced by the presence of the adjuvant. So the virus-like particle after a single dose generates relatively little neutralizing antibodies, which is significantly boosted by the second dose. But there's a very clear advantage to having either of these adjuvants for dose 1 or dose 2. In the induction of neutralizing antibodies, and this is measured by the VSV pseudovirion assay. Next slide. Our very preliminary data from the ELISpot from this mouse study suggests that both the VLP by itself in green, as well as the VLP in the presence of AS03 or CpG, is able to induce a gamma interferon ELISpot type response. that there are more data coming every day. This is what I had at the time of sending the slides into ISV. Next slide, please. We, like many others, are running multiple studies at the same time. So as the mouse study is being done, we have a large 44 new non-human primate study ongoing with the help of New Iberia and Tulane in the United States. 44 Indian rhesus macaques received 1 or 2 doses of our vaccine at 15 micrograms, which is the highest human dose we plan to use alone or with ASO3 or CPG. Some of the animals were sacrificed after a single dose, and other animals are were just sacrificed this past week after 2 doses. 28 days between the last vaccine and sacrifice. Some of these animals are also being challenged. I'm sorry, I misspoke. A small number of animals are being sacrificed within 3 days of the challenge, and the remaining animals are being sacrificed 14 days after challenge. Next slide, please. These are the outcomes that we are looking at in the animal, in the non-human primate study, where we're doing standard safety tests, bronchoalveolar lavage, swabs, obviously, to look at virologic outcomes, clinical signs, and then sacrifice at 2 time points after challenge. The immunologic outcomes are being done in 2 waves. We're looking at standard immunologic outcomes, the same ones that I mentioned in the mouse study, and those are being done with a combination of collaborators through the United States, including Bally Polendran at Stanford, and then a second wave of more sophisticated systems vaccinology type assays that Dr. Polendran is helping us with, which will be coming as a second wave. Next slide. So far, in the non-human primate studies, we've seen no evidence of enhanced disease, either with worsening of clinical symptoms, with X-rays, and we are waiting on the immunologic outcomes from these animals. But to date, we've seen no evidence of vaccine-enhanced disease, at least with the short-term challenge at either 3 days or 14 days after challenge, after 1 or 2 doses. Next slide. And go forward just one more, just to show you where we are. You are here now. This is where we are now. We've actually got our non-human primate study running in the United States, and we've started in mid-July our first in-human study, which is fully enrolled within 180 subjects, where we are testing 3 dose levels of our virus-like particle, either alone or with these 2 adjuvants that we have partnered with GSK and Dynavax for. We are at the point where we have given almost all of the subjects their 2nd dose, and our non-human primate data and our human data will be emerging and analyzed over the coming 3 to 4 weeks. We will be using these data. We have submitted our IND to the FDA on the 7th of August, and we are planning actively for our phase 2/3 study. And my last slide is an outline of our clinical program. If you go to the last slide, please. So our phase 1 is dose finding, safety, immunogenicity in seronegative 18 to 55-year-old individuals, looking at the 2 adjuvants, 3 dose levels, prime dose, so 2 doses. We're evaluating the immune responses after the 1st and the 2nd dose. And in fact, this is fully enrolled. These individuals will be followed for safety for 1 year as planned. Instead of doing a phase 1, phase 2, as Sanofi has planned, we are completing the phase 1 and then choosing the adjuvant and 1 or 2 dose levels to carry into our phase 2 study, where we plan to enroll between 1,200 and 1,600 individuals to validate that we have the right dose level. In our phase 2 part, we will be including children and adolescents 5 to 17 years old, the elderly over 65, and individuals between 18 and over with comorbidities in a staggered fashion. The 2 formulations that we move forward, possibly 3, but we're planning on 2 right now, will be chosen from the phase 1 data. And as we generate more data during the phase 2 portion, that will define what formulation we will take forward into our phase 3 study, which will be event-driven either as a standalone or in collaboration with one of the consortia that are moving forward in the United States or globally. And the N for these studies, as you know, is between 20 and 30,000, with an expected duration of at least 6 months, with follow-up for vaccine-enhanced disease for at least 1 to 2 years. So we're at a much earlier stage of development, but we're moving very rapidly. And we hope that we will be able to release our non-human primate data and our early phase 1 human data sometime within the next month or 2. Our current plan is to start the phase 2 portion of our phase 2/3 study in mid to late October of this year, with a transition to the phase 3 portion of that study either in late December or more likely early January. Next slide, and is simply to say thank you for your attention. And obviously thank you to all of our partners moving forward to this date.
Well, thank you for a really interesting talk. I'm hearing an echo. I hope that you're not hearing it. So my first question for you is, what is the advantage of using plants as a platform for VLPs, and what are the challenges as far as the regulatory agencies go?
Well, I'll take the second part first. To date, we haven't had any real challenge with the regulatory agencies, in part because there's really no overlap between adventitious agents that infect both plants and human beings. So we have a significant advantage there. The plants, at least this relative of the tobacco plant, Nicotina benthamiana, is very efficient at producing proteins in their leaves. And so we actually have fairly high yield. So instead of using a large stainless steel bioreactor, we can actually do all of the upstream bioreactor work in a greenhouse and then move the material, the biomass, into a relatively small and contained downstream purification procedure, so that in fact we can we actually can produce large amounts of vaccine with a relatively small footprint, a relatively small GMP typical footprint. We obviously have the greenhouses on the outside of the GMP facility.
Thank you. Can you tell us how far your flu vaccine is?
Our flu vaccine is currently, it has been submitted to the Canadian regulators, and we are in, we hope, the final stages of seeking the licensure for use in Canada, and we are in continued discussion with the FDA.
Yeah, well, so I'm just looking to see what questions, it's a bit difficult to figure out the questions here. There's a question on when the vaccines will be tried in children instead of adults.
So the vaccines, we currently plan to enroll children in a second wave of the phase 2 portion, the phase 2 portion of our phase 2/3. So starting, we hope, in mid to late October, we will have identified the 2 dose levels that we want to analyze. Once we have recruited a larger number of adults, 50 to 100 healthy adults, we will have a data safety monitoring committee that will, we hope, give us permission then to start enrolling initially the adolescents, so 13 to 17, followed by the younger children, 5 to 12. So late 2020.
Yeah, and my last question, which I'm sure you always get, is what about anti-plant antibodies?
So that's a really good question. To date, we've excluded people who have anaphylaxis, documented anaphylaxis to plant materials. But we have actually included people with seasonal allergies and non-anaphylactic type plant material allergies in our studies to date, and we've had quite substantial numbers of people enrolled in our studies and seen no evidence of either the induction of or the worsening of these plant type allergies. Furthermore, we've actually looked at the IgG and IgE responses to the plant glycans that are known to be associated with the severe reactions like anaphylaxis, and our plant-made VLPs do not induce those types of anti-glycan IgE responses.
Well, that's very reassuring. But thank you for a very interesting talk. I really enjoyed it, and I wish everyone who's presented a speedy path to get us vaccines. We need them. Thanks a lot.